let's get into it because we have Lance waiting for us. So we have seen so far in this study weekend that we have like an extreme, the last year's extreme increase in structures that has been deposited, and this development had led to all these really nice tools, mostly based in machine learning, that can give us like expand our capabilities in the field. And that was really nice, really revolutionary. But there's still some stuff that we're lacking, and I know that we don't understand exactly, and these tools cannot like provide answers just yet. And one of these things is like how we can understand better the dynamics of the protein. So how we can get from one conformational state to another. And this is important because proteins are not static entities. So we have like, in order to fold, in order to like have enzymatic catalysis or like allosteric regulation, this is all like functions of the protein that we're interested in, and they governed by the protein dynamics. So can we use the growing number of the static structure that we have already used to create these really nice machine learning tools to also understand the dynamics, not just stay to the static structures? And just like to clarify, I'm meaning computationally, because of course we have like experimental methods like time-resolved crystallography and so on that can help us with that. But what about like the computational part of things? So in our team, we have developed this software that's called ROPE, which stands for the Representation of Protein Entities, and is a torsion representation of the protein instead of an atomic one. And this allows us to explore the conformational space of the protein based on the structure that have been deposited on the PDB or a structure that you might have solved. So I'm just going to talk to you a bit about ROPE, and then like how I combined ROPE with some other methods in order like to go into the direction of understanding the dynamics. So very quickly run through of how ROPE works, and like if someone has questions of that, we can discuss later. So ROPE, in ROPE, the only thing that you need to start your analysis is to have a reference sequence. So like you are interested in a protein, you have the reference, you enter it into ROPE, and then ROPE can go and find all the PDB structures that are available in PDB that are similar enough to the reference sequence. Then it extracts the atomic position and goes into refinement using our own refinement method called Vagabond, which is a torsion angles refinement method. And the reason that we go with torsion angles representation is because it allows us to represent the system with fewer parameters, and it's easy, sorry, it's easy to interpret. So then after we have the refinement, we go through some like behind the scenes steps, let's say, and in the end we end up with a correlation matrix to which we can find the singular we can apply the singular value decomposition, and in the end we will have a view of the static structure conformational space. And I'm going to show you in an example what does it look like in ROPE. So let's assume that we have a structure. We want to study the structure of hemoglobin. Of course, like it's one of the most well-studied proteins, and we have a lot of structure out there. For example, here you can see like I found from the reference center that I entered, I found 244 models in the PDB with 98% sequence homology, and this is how it looks in ROPE. So like each dot here represent one observed conformational in rope space. So like if I go into one of those, I can see that one of them like represent a model in the PDB. And so from then on, that's like kind of initial thing that you can see like where the structures lay, and that you can go on and like have more analysis into rope. For example, it's, you all know here better than me that the hemoglobin is like it has two main states. You have the T state and the R state, and you can go on rope and color and ask to color if you know like two residues in your protein that it's a specific interest, and you know like these chains from one state to the other, you can go and model your structure based on that, and you can see a very clear classification of like most, not all, but most of the T state structures lay in this side, like closer to each other, but more spread, and there are states like more cluster in this side of the conformational rope space. So this is kind of an introduction on rope, and I want to focus in a case study using the human gammatic crystalline protein. So this is a single chain protein, but in the unit cell, it's packed as a two chain X and chain A. So if I look into some experimental structures of this protein into the rope space, I have like 16 PDB models, and it looks something like that. So like the chain X lies in this side and chain A lies in this side. And like this protein, each chain has kind of a different movement, let's say. And so if I look, if I calculate this plot, which can be like seen as a map of the relative distance between each atom, each CA atom in the chain, it looks something like that. And like from here, like the more yellowish, reddish to yellowish the regions are, the more farther apart the atoms go, and the more bluish to like dark blue, it's like the most closer they get. So I can see from here, I can interpret this as like the chain X in this case. I have like this movement of like moving closer to another, the domains moving closer and away from each other. So I can, from this, I can have like an idea of how the protein is moving. But so here, like I saw you an example with hemoglobin, which I have like hundreds of structures available, and I give you an example of this, which I have 16, which is like much less, but still can produce some results. But what we have, if we don't have, I only have one structure, and I want to see what the conformational space looks like. So there is ways to do that, of course, like the dynamics, like as the two previous talks discussed. Of course, you have MD simulations, very accurate, very versatile, but they come with a cost of time and size a lot of times, and the force field limitation, you don't know exactly, and so on. And then you have other methods like the elastic network model. And the one that I would like to talk about next is the kinematic flexibility analysis. And we came across in this method, and we thought like, oh, this is a nice way to mathematically describe and make sense the flexibility of the protein. And we want to see if we can, first of all, verify the results that we're getting for our test case. And second, like see if we can natively implement it in rope and what that will give us. So I'll just like very briefly go through like explaining the KFA method, kinematic flexibility analysis, which you can find more details on this paper. So starting from a static protein structure, I can represent the protein as a multi-body of complexes with torsion angle as the degrees of freedom contributing to the molecular motion. So like looking here, each one of the cycles represent an atom, and then I have categorized the different bonds into direct rotating bonds, single covalent bonds, double covalent bonds, and hydrogen bonds. And each one of these bonds will create a rigid body in the protein. So, for example, its color here represent like a group of atoms that are rigid inside the protein. So here, for example, the green one, like it's a rigid group of atoms in there, and the purple one as well, but there is movement in the torsions between them. So then I can do, I can also represent, from there on, I can also represent the protein as a kinematic spanning tree. So each one of these, so instead of now having the atomic representation, I have the rigid groups and how these rigid groups move with each other. And so the vertices here represent the degrees of freedom, and the edges represent the holonomic constraints. And I can also have some other, which is important in this part, some other bonds, which is the hydrogen bonds, which is the only ones that allowed, not the only ones, the ones that allowed the more variation on the distance between the groups. So, okay, this is, I wanted to introduce kind of like a very simple example of like more intuitive. So imagine instead of a protein that we have a marionette, that you have the hinges represent the torsion angle, like the torsion angles in the protein as the degrees of freedom against, and the connections as the distance constraints. So like, then like a marionette can like, the distances between the hinges cannot change, but the rotations is how much they can rotate, they can. And, but like, I can introduce a wooden leg, let's say, in the marionette that will constrain the movement between the knee, like the knee. So like the knee cannot move now. So the movement will look something like that. So it will be different from when I didn't have, like each constraint that I add, it's correspond to a different kind of movement. And so like I can also add another constraint, another kind of constraint, which is now, let's say, a spring that instead of just like restricting the movement, like it allows like kind of variation in the movement, but in a, like not, oops, sorry, not the crazy variation. So like you can go farther away and change the distance, but after a certain level, this like constraint will break. So so all possible configurations that the margin can take while satisfying the constraints that I added will lie in a surface that's called manifold. And you heard about manifolds in the previous talk again, but let's say that each one of this configuration is like a dot, like a dot in this like space, that's like in the 3D space. And I know, and everything I need to know about how the protein will move, it's in this, lies in this manifold. If I only could access the motions, extract these motions from the manifold. To do that, in mathematics, we usually use this, what we call the tangent space, which represent the very, 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 very small motion available for each configuration. So for the marionette, this can mean that a small movement of the limbs that satisfy the string length constraints. For a protein, this is equivalent to a small adjustment in the backbone that present the structural bonds. And in order to encode that, we, again, like in physics and mathematics, we can calculate a map, let's say, that gets you from the manifold to the tangent space, which is called the Jacobian matrix. And it looks something like that. So you have the degrees of freedom as columns and the constraints as the rows of the matrix. And then we can determine the singular value decomposition of the Jacobian matrix. And then we have three different, we decompose the main matrix into three different matrices that is more easy to interpret. And we have the null space. One of them is the null space, correspond to the null space, which means like the matrix that highlights the direction where motion is unconstrained, sorry. And the column space, which is highlights the direction where the motion is constrained, is so more rigid. So that's the theory behind the KFA. And now I'm going to move into showing you how you can use it with rope and what result we get. So the way I use it here is like to create kind of, let's say, an example of starting for one experimental structure, an example of like of protein structures that they have just like moved in a tiny bit manner in the flexible regions. So I created the 50 sample structure for this example. This is like the crystalline that I showed you before in rope. So 50 sample structure originate from one experimental PDB, and the rope space, the conformational space looks something like that. And this is like just, I should know that this is like only train X this time. So I did the kind of the similar analysis as I did with the experimental structures and with the sample structures with, ignore that. So now I can see that the KFA sample structure in the single chain and the experimental structure, this distance map, let's say distance difference map, is very, very similar, which gave me a very good indication that the way the KFA produce these samples is correct in the sense of like it can be nicely reproduce the experimental results. So that was a good indication that we're going towards the correct direction. And then we wanted to also have everything in one thing, because until this point I was using, to sample the structure, I was using the algorithm from the original paper. So we're now moving on like to implement everything on rope, and I have implemented the like I now can calculate the flexibility inside rope and have this kind of create this kind of movement, let's say, where when I move the bar, I can see kind of the movement of the protein, and it's the movement that I'm expecting, and like the flexible region is moving where I'm expecting for them to be moving. Yeah, so that's all for me. I think that we generally, I think this project is in the beginning. There's a lot of things that we want to dive deeper into. Like, it will be nice to see if we can predict or calculate anisotropic B factors and see how they compare to the experimental ones, and explore the impact of the hydrogen bond network on the torsion network in the flexibility. And yeah, as a main output, I would also like to mention that it's a nice way, this project, like to combine different, like a bit of mathematics, a bit of physics, like the whole rigidity theory. It comes like it's applied in robotics. It's someone could say completely different what is crystallography, and I think it's a nice example of like getting inspired for other fields and combining together to have a different perspective on a subject. Yeah, that's us. Yeah, thank you.
Thanks very much for the talk, Virginia. Are there any questions in the room? No. No. While other people are thinking of questions, I'll ask one of my own. So you showed that from one structure, you were able to fairly accurately reproduce conformational space that seen experimentally.
Yeah.
How many different proteins have you done this for? And have you been able to show that you're able to reproduce kind of large conformational changes?
No, I think that this can be I can be quite sure that this is only for small conformational changes. And you can see that when you calculate the sample structures and you see them in compare with the experimental structures, that they're very, very close by cluster around the original structure, which means that they don't really have large variation, conformational variations. And unfortunately, I haven't tried, I have tried a few more proteins. It really depends of, I have some examples that they're not so rigid, and that's why I try the example, because I know that it's more they're more rigid, less flexible proteins, and I have tried in some I can see some good encouraging results, but yeah, it's kind of new, so we still have some. I need to try more proteins, basically. Yeah.
Okay, thanks. Yeah, we've got a question up there from Alan.
Hi, Virginia. Thank you. Alan Orville, Diamond Light Source. This question about limited number of proteins in the dynamics. If you have a small protein, but it's in multiple unit cells, different crystal forms, it might give you, or could it give you differences in the dynamics that you predict? In other words, what's the impact of crystal packing for your analysis?
I think that this example with the crystalline, it's really nice because in general, you have two chains in the unit cell. And when you analyze just with ROPE, the experimental structure, you're looking into chain X in relevance of chain A, and you just see the movement in chain X. And then when I did the same analysis with the sample structure, I just used chain X, and I could see that just by having chain X, the mathematics and the physics behind the KFA method was well enough to reproduce this movement. So that's, I think, an encouraging example that probably it's well enough defined, the KFM method, to actually reproduce the motions in multiple packing.
Any more questions in the room? No, in that case, if we thank all three speakers again, and